Okay, everyone. So let's continue with our discussion in chapter 14. Yeah, that's where we are, chapter 14. And when I left you, we were talking about our boy, Gregor Mendel, and about all the wonderful things that he discovered. And let me just remind you that Gregor was doing his work in the late or mid 1800s. So he began somewhere around the 1850s. A lot of folks will say 1856 or 1859. He published his work in 1865. Now, at this point, we don't know anything about genes, and it wasn't really until late in the 1800s that we had good enough microscopes that we were able to observe meiosis at the cellular level. So all the things that Gregor figured out, he figured out without the benefit of looking at the cells themselves. And I have some bad news about our buddy Gregor, and that is that his work was largely ignored for a long time, for several decades, because it was published in a sort of obscure journal. He was just this monk in the Czech Republic. And so no one really noticed it. Not until 1902 to 1903. In those years, something important happened. And that is that there were two scientists that were looking at cells through microscopes. One was named Dr. Sutton. I cannot remember his first name. And I think the other guy, I know his last name is Bavary, and I think it's Theodore Bavary. But these two dudes were cellular biologists. They were observing cells through the microscope, and they were able to observe meiosis. So they were actually able to visibly see chromosomes. See chromosomes during meiotic division, okay? So what these two dudes did is independently, they came to the same conclusion. They had this idea that chromosomes carried the genetic material, okay? It was the instruction book, okay? So genetic material. On the chromosomes. And they rediscovered Mendel's work and realized that Mendel's work could be explained by the pattern of chromosome movement that they observed in meiosis, okay? So here, for example, they noticed that, you have these two chromosomes. These chromosomes duplicate during the S phase. They then separate during meiosis 1, and then the copies pull apart during meiosis 2. So they recognized that this would explain Gregor's principle of segregation. Here, we have a situation where you have the paternal chromosome and the maternal chromosomes lined up on either side of the cell. So if you undergo meiosis, you end up with a particular variety of alleles. Here we have big R, big Y, little r, little y. This should sound familiar from the previous lecture. But if the chromosomes line up differently, then you would end up with a different assortment in the resulting gametes. Remember that this is the principle of independent assortment, and that arises because these chromosomes here line up in a random fashion. The father chromosome and the mother chromosome, whichever one is on the left and the right, that is completely random, which means that you can end up with a random mix of alleles, okay? So going back to these guys, what they are credited with is called the chromosome theory of inheritance. And they got there by rediscovering Gregor Mendel's work and observing meiosis. Now, a little bit of terminology. One of the things that they figured out was they were the first folks to decide or to suggest that these hereditary factors were something called genes, and that these genes resided at specific locations on chromosomes. So for example, we're saying that this gene for seed shape resides somewhere up here on this chromosome. And then maybe down farther, there are other genes, and maybe there's another gene here and another gene here. So a single chromosome has many, many genes on it. And there's a specific location on the chromosome where a gene resides. That specific location is called a locus. Okay? Multiple locations, so plural, are called loci. And that's just Latin for location. Okay? So in 1903, 1902, we now have some visual evidence that explains Gregor Mendel's work. But at this point, we had no direct evidence, no experiments to confirm this idea that there were genes on chromosomes and that these chromosomes carry genetic information. So when do we start getting some evidence? Well, not long after the chromosome theory of inheritance was decided or was proposed, folks started looking for these chromosomes and looking at ways to study them. And one of the animals that people decided to work with was a very famous model organism called the fruit fly. Now, the scientific name for the fruit fly is Drosophila melanogaster. And you'll often hear people just abbreviate that, Drosophila. This is a really important model organism. A lot of genetic work has been done and has been figured out using these simple, humble little fruit flies. Why are these such a great organism to study if you're studying genetics? Well, for one thing, they are super easy to rear in the laboratory. You probably have had these in your house. You brought a banana or something in and you've let it get all brown and icky, and you've seen little flies hovering around it. And if you've ever gotten these fruit flies in your house, they're super hard to get rid of because they just kind of grow and grow and grow. The other thing about them is that they have a really short generation time. I mean, we're talking like just a couple days. So in a period of just a few days, you can have like a whole new generation. So you can go through multiple generations of fruit flies in a week. Perhaps one of the features that makes them one of the best models we could study is that they only have 4 pairs of homologous chromosomes. And these are big chromosomes that are super easy to see under the microscope. So here we go. Here is an example of the chromosomes of Drosophila melanogaster. And if you look, first of all, notice that these are unreplicated chromosomes, right? They do not have that classic X shape that you would expect if they had gone through the S phase of the cell cycle. But you can see that this is a homologous pair, this is a homologous pair, this is a homologous pair. And believe it or not, that is also a pair of chromosomes. Now, what you should probably figure out is there are less genes on this chromosome than there would be, say, on this chromosome, because frankly, there's just less DNA. There's less real estate, if you will, for those genes to reside upon. Now, folks began using these fruit flies in the early 1900s, and a number of scientists began working with them. And one of the first important discoveries that was made using these fruit flies was made by a female scientist named Nettie Stevens. And I bring her up because I'm going to talk about another person in a minute who's really famous for pioneering fruit fly work. His name was Thomas Hunt Morgan. But honestly, without the work of Nettie Stevens, Thomas Hunt Morgan could not have done his work. And as is the case with many minority scientists, her work hasn't really been highlighted in the past. But what Nettie discovered is that in female fruit flies, their chromosomes look a little bit different. Okay, let's look here at this female fruit fly. Looks like we have 4 homologous pairs of chromosomes, right? Now let's look at this male. These 2 chromosomes look the same. These 2 chromosomes look the same. These 2 look the same, but these 2 look quite different, don't they? So she was the first person to really propose that there are some chromosomes called sex chromosomes that determine the biological chromosomes, that determine the biological sex of an organism. So here, the presence of this Y chromosome makes this a male. In the absence of that Y chromosome, you are not a male fruit fly. The same is true in humans. If you lack a Y chromosome, you are not biologically male. Now, that's a whole different conversation from the gender conversation that we have today about people's sex or gender and how they identify. We're talking about at the molecular level. So just to recap, these chromosomes here that are not involved in sex determination, these are called autosomes. And in contrast, the chromosomes that are involved in sex determination are called sex chromosomes. Now, Nettie's work really enabled the work of another scientist. Let's see here. Oh, I put this on here, sorry. Just to show you that human sex chromosomes are also present. This is an example of the human X and the human Y. Human Y is much smaller than the human X, and therefore it does contain fewer genes, but its presence does confer biological maleness on an individual, okay? So if you are biologically male at the cellular level, then you can see that meiosis would produce two different types of gametes for you. 50% would contain an X copy, and 50% of your gametes would contain a Y copy. And so this is all just to show you that it is the male. Biologically, it is the male that determines the sex of the offspring, right? Now, why do I say that? Okay, why do I say that? Because if you're a female, all that you have in your cells are two copies of the X chromosome. So when you undergo meiosis, and we're not going to go through all the steps, but you can see that you would end up with 4 copies of an individual X chromosome, right? So the differentiator is here in 50% of the male gametes. Okay, back to fruit flies. So when Nettie had completed her work, it really enabled the work of this dude named Thomas Hunt Morgan. And I want to say that he was working in the 1920s. For some reason, the year 1929 sticks out in my memory, but I'd have to review that. But what he's really famous for is pioneering the use of the fruit fly. So we'll just put he pioneered. Why can't I spell this morning? Pioneered fruit fly genetics. He had an entire lab where they just bred pioneered fruit genetics, fruit fly, genetics. He had an entire lab that was dedicated to breeding these dudes. And what he was hoping to do was to create some mutants that he could follow, because it turns out that these fruit flies were not domesticated. And so they didn't have all these interesting features like the peas that Gregor Mendel had. So there wasn't sort of like pink and white flowers or yellow and green seeds or wrinkled and round seeds. Pretty much what you had was a series of flies that all looked really similar to one another. And the characteristics that you find in an organism that lives in the wild, those are often referred to as the wild type characteristics, okay? And often, although not always, these are dominant features, okay? Not recessive, okay? So what Thomas Hunt Morgan and his colleagues in his lab were trying to do is, through the process of breeding so many generations upon generations, he was hoping that a random mutation in the DNA would occur that they could follow as they mated these fruit flies to try and understand a little bit about fruit fly genetics. And lo and behold, at one point during their breeding experiments, they found a mutant that they could visually identify. And this is an example of the mutant here. This is a white-eyed Drosophila. Notice that this normal wild type Drosophila has a red eye, okay? So white is a mutation. And so they were like, great, we want to provide some experimental evidence for the chromosome theory of inheritance. So we're gonna do some crosses between red-eyed flies and white-eyed flies. And so what they did is they had some pure breeding red-eyed flies and they mated them with a white-eyed fly. Now, let's look at what that might look in terms of a Punnett square. And we're gonna use W only because that is the letter that's been used traditionally for this trait. Doesn't make much sense to me because red is the letter R, W is for white. But anyway, we're gonna use a capital W to equal red and a lowercase w to equal white, okay? Now, what Thomas Hunt Morgan and his colleagues assumed was that red was probably dominant because that was what was out in the population, and that this white mutation was probably recessive. So what would happen if we took a pure breeding, remember that is code for being homozygous, a pure breeding red, and we matched it with a recessive white. And remember, there's only one way to be recessive. You have to be homozygous to exhibit the recessive phenotype, because remember, if you were heterozygous, your phenotype would be dominant, right? So you know if somebody has a recessive phenotype, you already know what their genotype is if they are recessive. If they are dominant phenotype, you don't know if they are this or if they are this. But the good news is I've already told you that they established a pure breeding line of red eyes. So we know that we have homozygous reds and homozygous whites. So here is what we would expect. What we would expect if we made it a white fly. This would be a white female, okay? And this is the symbol for female with, a red. A red female, let me write that down so I don't confuse you. A red female with a white-eyed male. Sheesh. Oh, here we go. White-eyed male, boom. And this is a male. Okay, what you would expect, oops. This is what you would expect, is for all of them to be red, right? This is your expectation. Well, guess what? That is not what Thomas Hunt Morgan found. He found that the females were red and some of the males were red, but there were a few that were white-eyed, and those were always males that were white-eyed. So what he discovered was something called sex linkage, or sex-linked traits, okay? What is happening in a sex-linked trait is that the gene for eye color is on the X chromosome. If you are a male, you only have one copy of that gene because your other chromosome is a Y, okay? So if you are a male and you have the white-eyed trait, then you give one copy of your X here, which is the white-eyed copy, and one copy of your Y here. Here, we have a female that has red, red. She donates an X with a red. So you have an X with a red gene and an X with a white gene. That is going to be red, right? And then here you have an X with a red gene and a Y. That's also going to be red, okay? And I'm going to do this again so it makes sense. Now let's switch it. Let's take a wide-eyed female, and you're thinking, how are we going to get that? We'll get there in just a second. Let's take a wide-eyed female. It is possible to get one of these, and we will talk about how. And let's cross it with a red male. So this red male has an X chromosome with the red gene on it, okay, right there, and a Y chromosome. Here we have a female that has 2 copies of the X chromosome with the white gene on it, so that's all she can get. So in this cross, you have a female that's red because we have a dominant and a recessive, and you have a male that's white because you have recessive and a Y chromosome. I'm not sure if this makes sense, so let's work it out in a larger Punnett square example, okay? Let me skip. Okay, let me do this. Okay, so let's start out with what Thomas Hunt Morgan started out with. He started out with a red-eyed female. Oops, sorry, there we go. A red female. Okay, I'm gonna draw it like that. And a white male. All right, let's draw this out. Now, actually, let me make this a little lower because I'm going to need some space. Now, the way that we typically represent sex-linked traits in a Punnett square is by first determining what the sex chromosomes that would go into the gametes would be. So if you are a red female and you are true breeding, I'm going to put TB here for true breeding. We know if you're female, you have 2 X's. This female is true breeding. She is homozygous dominant, meaning she has 2 capital W's to make her have a red phenotype. Let's look at our male. If you are male, you must be X and Y. There is no place on the Y chromosome where this eye color gene resides. The eye color gene is only on the X chromosome. So in order for this guy to have white eyes, he's got 1 copy of the X and it's a little W. So now let's put all this on our Punnett square and work it out. Okay, so I am going to put, I'll have to make it small here, an X and an X and a big W and a big W. That's the mom. And I'm going to put an X with a little W and a Y. That's the dad. Okay, so here, if the gametes that fuse are these 2, you would have a female, so 2 X's, and you would have a big W and a little W. This individual would have red eyes. I hope you guys can see that. Okay, let's look at this individual. So here we're going to have an X and an X. That is a female, right? And we're going to have a big W and a little W. That is a red normal female. Okay, now let's move down here. If these gametes fuse, you're going to have an X and a Y. Now you're going to have a male, and on this X chromosome, you're going to have a red allele. And on this X chromosome, you're going to also have a red allele. Interesting. So all these are red. Now, let's take one of the females from this cross, okay? And let's cross it back to one of the males from this cross, okay? So let's draw a second Punnett square. So here's our female, X, big W, X, little W. And here's our male, X, big W, and a Y with no eye color gene. All right, X, X, female, big W, big W. Hopefully you can tell that that is red. X, X, big W, little w. Oops, that is a terrible little w. Hopefully you can tell that that is a female that is also red. All right, X, big W, Y, that is a male that is red. And then here we have an X and a Y, but check it out, little w and the Y. This is a white-eyed male. So you can see that what's happening is because a female has 2 copies of the gene, it is very rare for her to exhibit this trait. But males only ever inherit 1 copy. So if they get a defective copy from mom, that's what's going to show up. In this case, where someone is carrying a good copy of the gene and a defective copy of the gene, we call them a carrier. Okay, so this female is a carrier for the white-eyed trait. Now, what's interesting is that this provides evidence for the chromosome theory, okay? Because we were actually able at this point to observe a gene, a trait. We knew that it had a location on the X chromosome, but not on the Y chromosome, and we could see that it followed inheritance patterns. So that is very, very cool. Now, here's a question. I'm telling you that this sex-linked trait is more common in males than females because males only inherit 1 copy of the gene. So my question is it possible for us to get a white-eyed female? And the answer is yes, but only under a very specific set of circumstances. So how can we get a white-eyed female? But how get a white female? All right, maybe you've already figured this out. Now remember that to get a white-eyed female, what are you going to have? You're going to have 2 copies of the X chromosome. Both of them are going to have the defective little w gene on it. Okay, so that means you're going to have to get 1 copy from dad and 1 copy from mom. So how could that happen? Well, let's just say that you had a mom who was a carrier and you had a white-eyed dad. All right, here we go. This is a normal female. She's red-eyed. But looky look, right here, what do we have? Little w, little w. Here is your white-eyed female. Here is a normal male. And looky look, here is a white-eyed male, okay? So yes, it is possible to get white eyes if you are a female, but it is less common than it is in males. It is more common in males. Now you may be asking yourself, oh, is this just a phenomenon that is restricted to fruit flies? No, there are a number of traits that in like humans that are inherited this way because again, in humans, you also, as a female, have 2 copies of the X and only 1 copy of the Y. Probably the most classic example of this is color blindness. Okay? The gene that makes people blindness, the gene that makes people colorblind occurs on the X chromosome. It is much, much more common in men, but as you can see, it is also possible to have color blindness if you are a female. So we now have evidence for the chromosome theory of inheritance, but we also have a little bit of an expansion of Mendel's rules, if you will, right? This isn't really anything that Gregor Mendel really discovered. So let's talk about some ways that genetics might differ from the basics that we discovered with Gregor Mendel. Let's see here. Oh, okay, other ways that traits don't follow Mendel's rules. Yes. Okay, so the first thing that I want to talk about is what if the genes are on the same chromosome? Up until now, we have been talking about traits that occur on different chromosomes. So if you go back to when we talked about round seed shape in peas and yellow color, and I was showing you how those alleles segregate during meiosis to form gametes. What I was showing you is that for each trait, each gene, it occurred on a different chromosome, didn't it? So when the chromosomes moved and ultimately combined to form these gametes here, we have all of this recombination, okay? You can, like I said, you can have RR, big Y, little y, and you can have this combination or that combination or this combination or that combination, because these are all on different chromosomes. But remember me telling you that each of these chromosomes, if you go back, each of these chromosomes has a whole bunch of genes together. So when you get this chromosome from dad, you're getting all of these genes together as a package, okay? So here's an example of that. If we're talking about fruit flies, two genes that occur on the same chromosome are gray body and white eyes. So here, this gets inherited as a package, okay? Gray body, white eyes. Here's a yellow body and a red eye. This gets inherited as a package. There are only two types of gametes you can have, the gray-white gamete or the yellow-red gamete, okay? That is different than what we learned before. What we learned before is that if you had, let me see here. Let me not do that, I don't want to confuse you. Okay, so you can have two different gametes here instead of the four, okay, that I showed you here. Remember, here you can have four, big R, big Y. Hopefully this is making sense. Here, these are together. So this is called linked genes, okay? Do not confuse this with sex linkage, okay? I know that's confusing, but they're linked in that they are inherited together as a chunk because they're on the same chromosome, okay? Now, I said that they're inherited together because they're on the same chromosome, but there's one exception. Sometimes genes on the same chromosome are not inherited together because if you recall in meiosis, there is something called crossing over. Crossing over is when the legs here get tangled up, and sometimes they can swap genes. So let's look at that on a slide. So here, can you see that their legs have become tangled up and these two have swapped genes? So you have, instead of the two options that are normal with genes on the same chromosome, you actually end up with four options that result from crossing over, okay? So in general, linked genes are inherited together unless there's been a crossing over event during meiosis, all right? So far, ways that our traits don't follow Mendel's rules, we've talked about sex linkage and we've talked about gene linkage, and do not confuse these two. Another way that traits don't follow Mendel's rules is something called multiple alleles. So far we've talked about things like red and white eyes, or yellow and green seeds, or round and wrinkled seeds, right? So you as an individual can only have two different alleles because you have two different chromosomes, right? You can have an allele from mom and an allele from dad. But sometimes out in the population, there are multiple alleles. And the best example of this is blood type. Now let's think about blood type for a second. You can be blood type A, you can be blood type B, you can be blood type O, or you can be blood type AB, okay? If you're blood type A, there's two ways you can be blood type A. You can get a copy of the A allele from mom and a copy of the A allele from dad, or you can get a copy of the A allele from one parent and an O allele from another, because O is recessive. So let's look at B. B, you can get two copies of the B allele from mom or dad, or you can have a copy of the B and a recessive O. Okay, so that's how you can be B blood type. Let me erase that comma. To be type O, there's only one way to be type O because the O is recessive. And what's interesting is because both A and B are dominant, if you get one copy of A and B from mom and dad, then you are blood type AB. So as individuals, we can only have, let's put here, individuals can only have two alleles, but there can be multiple alleles in a population. And this phenomenon is called multiple allelism, okay? So let's see here. Ignore this for now. This is essentially what I just told you over here, but I think it's a little more complicated, but it's essentially the same thing I told you over here. All right. The next rule that I want to talk about also can be explained by blood type, and that is something called codominance. Now remember that I told you that both the A allele and the B allele are dominant in blood type. So if you get a copy of A from one parent and a copy of B from another, you have a unique new blood type called AB. How can that be? Well, let's look at what's happening in this idea of blood type. What the A allele is coding for is a protein that's on the surface of your blood cell. And this protein has a specific shape and a specific structure. And just to make things easy peasy, we call this the A protein. The B allele codes for a second protein, okay? And again, it's got a specific shape and a specific structure, and it exists on the surface of blood cells. You can see how here's your cell membrane. Here we go. Okay, so here we have a B protein, okay? The O protein, or sorry, the O allele codes for no protein here. So you can see how if you get an A allele, you're gonna end up with this on the surface of your blood cells. And if you get a B allele, you're gonna end up with this on the surface of your blood cells. But if you get an O allele, there is absolutely no protein there. So you would only be blood type A. Let's work through that one more time. Make sure that's, oops. Nope, what I wanted to do was erase you guys. Okay, so let's say that you had two copies of A from mom and dad. Well, that would be easy, blood type A. All of the proteins on your blood cells would look like that. Okay, now, let's say that you get a copy of A and a copy of O, one from mom, one from dad. Well, because there's nothing here, the only thing that's being expressed is A. You're still blood type A, okay? We could do the same thing for B. If you get two copies of this B allele, then all of the proteins on the surface of your blood cells are going to have this B shape. If you get a B and an O, then again, you're only going to have this blue one here, aren't you? But in a codominant situation, like in blood type, you're going to have both of these on the surface of your blood cells. So these are codominant, okay? Again, Gregor Mendel didn't observe this. This is a little different and a little bit of an extension of his principles. Now, similar but not quite the same as codominance is something called incomplete dominance. And this is a little bit easy to confuse, okay? So in incomplete dominance, what you have is a blending effect. So here you have red, here you have white, here we have a hybrid, okay, and it's pink. And then here you can see if you do your next cross, what you would end up with. So incomplete dominance is when you end up with a blending effect, kind of like our blending inheritance hypothesis, but this only happens some of the time, okay? And again, it has to do with things that are happening at the molecular or cellular level. All right, another example of how traits do not follow Mendel's rules is something called pleiotropy. Pleiotropy is when a single gene, usually a mutation in a gene, has multiple phenotypic effects. Okay, so here's an example. There is a disease called Marfan syndrome. And I have some pictures of people that suffer from Marfan's. Marfan syndrome is a defect in one particular gene called FBN1. And this one gene causes some problems with your growth and with your cartilage. And some of the effects that you get are really, really tall stature, long limbs. You get an abnormally shaped chest. I don't know if you can see in this individual how their chest is kind of sunken in. That does result in some cardiac effects. So this defect in this one gene causes multiple phenotypic effects. Some little interesting facts. We think, based on photographs of Abe Lincoln, we think he might have suffered from Marfan's. He was extremely tall. He had a very thin face, very long fingers. So we definitely suspect that Abe Lincoln had it. And some of you guys know that I'm a huge Star Wars fan. This is a picture of the Star Wars cast. This is the guy that played Luke Skywalker, Princess Leia, R2-D2, Han Solo. This is C-3PO, and this is the guy that played Chewbacca. His name is Peter Mayhew. He passed away, unfortunately. I was supposed to go meet him at an event, and unfortunately, he died before I got to do that. But yes, he had Marfan's as well. All right, another example. So we're kind of just going through a whole bunch of vocabulary here at this point, aren't we? Epistasis. Epistasis is when one gene masks or modifies another. And a great example of this is baldness, okay? So I've got that down here. One gene masks or modifies the other. So you can have blonde hair or you can have red hair. But if you have the gene for baldness, it doesn't matter what gene you have for hair color because your baldness gene is going to mask the effects of a blonde or a red-haired gene, okay? And again, if you take my genetics class, we'll talk about why this happens, but just for now, understand that epistasis is when one gene masks or modifies another. Polygenic traits is the next one I want to talk about. In a polygenic trait, what happens is multiple genes, so polygenic, affect a single trait, okay? So a great example of that is height. There's all kinds of genes that contribute to your height, as well as things like nutrition. So what that means is that these polygenic traits don't typically happen in an either or a binary fashion. That's not what they look like, right? We don't have tall, medium, and short people. We have people that range from being very short to being very tall. And that kind of results in a bell-shaped curve, okay? So we often call polygenic traits quantitative traits because they can be measured on a scale. Quantitative, yeah. They can be measured on a scale, okay? You might say intelligence is a polygenic trait because there's many, many factors that contribute to someone's IQ, for example. Skin color is a polygenic trait. So let me give you an example of that. In skin color, and we're going to talk about skin color as another example here in a second. In skin color, it turns out that there are 3 genes that contribute to skin color that we're aware of, okay? And these 3 genes code for melanin pigment production, right? So you can have a big D and that's a very dark skin color. And you can have a little d, and that's a very light skin color, right? But you have 3 copies of it. So if you were this, you would be a very, very dark skin colored person. And if you were this, you would have a very light skin color. But you can have all sorts of combinations in between that will result in various shades. Now, the thing about skin color is that it's also affected by environment. And this is something that Gregor Mendel didn't really factor in to his thoughts on inheritance, okay? So while you can have all these big Ds or you can have all these little Ds that sort of determine your base skin color, if you go out in the sun, you can change your base skin color, right? Because you can change your the melanin production by exposing your skin to the sun, okay? So let's see, what else do I have on my list? I would add as a number 9 here, environmental effects. The very last thing that I want to mention to you guys that's in this chapter is this idea of using something called a pedigree to trace inheritance patterns throughout families. This is done a great deal in the medical field or by genetic counselors to try and help people understand what traits they might be carrying that they could pass on to their offspring. So let me give you an example. This is an example of a recessive trait for a disease called sickle cell disease. And I think we probably mentioned it before, but sickle cell disease causes a deformity in your blood cells where they actually are shaped like little sickles or little letter C's, and they don't absorb oxygen very well, and they don't travel through your blood vessels very well. So as a result, it creates problems for people that are affected by this disease because they have less of an ability to oxygenate their tissues. They can have difficulty with exercising. They can have some problems with their organs functioning properly because their blood flow doesn't work right. So in a pedigree, what we typically see is that males are determined or are designated by squares and females are designated by circles. If you are what we call an affected individual, then your square or your circle is entirely colored in. So here's another affected individual. And if we're talking about a recessive trait, remember, we know that that is going to be a homozygous situation. So let's just say that we're going to use, I'll use big S to be dominant, and I'll use a little cursive S to be recessive. This individual is little s, little s. This individual is little s, okay? If we're trying to designate on a pedigree a heterozygote or a carrier is another way to say it, their box or their circle is half filled in, okay? So we know that this individual must be big S, little s. We know that this individual must be big S, little s, right? Big S, little s, big S, little s. Okay, so we can tell things by a pedigree. So for example, if you knew, for example, that you and your partner were both carrying the sickle cell trait, you could kind of look at how that trait is carried throughout your family history, or maybe you can work backwards. For example, if let's say that you have one affected offspring, right? And you're a genetic counselor and you're counseling this couple right here. Here's an affected offspring. You could be like, hey, the only way for you to have an affected baby is if both of you are carriers, and you might want to make a choice about whether or not you have more children, depending upon what disease you're talking about. This is another example of a pedigree. This pedigree is for an X-linked recessive trait. And how would you know this was X-linked? Well, here we have a male that's affected by red-green color blindness, okay? And so you can see that this male is affected and this female is unaffected. And the way that you can typically tell an X-linked trait is that the majority of the affected individuals are males. All right, this brings us to the end of chapter 14. And if you're watching this in the fall of 2020, this brings us to the end of the material for your exam. Again, if you're watching this in the fall of 2020, your exam will be on Friday, and it'll be during your normal class time. Good luck with your studying, and I'll talk to you soon. Bye-bye.